Ribosomes are very small organelles. They are made up of two subunits, one larger subunit, one smaller subunit. Okay. These are the organelles which are found in almost all organisms, whether it is a prokaryote, whether it is a unicellular, whether it is a multicellular, whether it is a eukaryote or prokaryote, what type of cell, plant cell, animal cell, in all these organisms you can find the ribosomes. So simply they are dot-like structures if you see. So where they are present? One, they are present freely in cytoplasm, they float in cytoplasm. Second, they can be found in the endoplasmic reticulum. They are attached to endoplasmic reticulum. Yesterday we have seen that those organelles, that part of endoplasmic reticulum which have ribosomes is called as rough endoplasmic reticulum. And then, third one. It is also found in mitochondria. It is also found in chloroplast. Mitochondria ribosomes are made up of 55 ribosomes, whereas cytoplasm ribosomes can be a 70 ribosome or 80 ribosome. What is S? S is Swedberg unit. To understand its dimensions, it is one unit that is proposed by the scientist Swedberg. This 70 ribosomes are present in prokaryotes. 80 ribosomes is present in eukaryotes. This 70 in 70 ribosome, we can see the subunits are made up of what is the subunit is made up of small. The smaller unit is made up of 30 Swedbergs and the larger unit is made up of 70 Swedbergs. 70 only. No, sorry, 50 Swedberg units. Okay. So coming together, it should be 80. Why is it not 80? Because this small overlap is present. So it comes only a 70 subunit. This is present in prokaryotes. But if you see in eukaryotes, the smaller one is made up of 40 subunits and the larger one is made up of 60 Swedberg units. Comes together to be 80 Swedberg units because of this overlap. Right? What are the subunits of mitochondrial DNA? Still it is under research. We will know in future. Right? What is the function of this ribosomes? Ribosomes function is very simple, protein synthesis. Wherever they are present, it is useful only for protein synthesis. In cytoplasm also, it helps in protein synthesis. Endoplasmic reticulum, I said you, rough endoplasmic reticulum is useful for protein synthesis. Mitochondria and chloroplast are very important organelles. There a lot of proteins are required. Why they require a lot of proteins? We'll discuss now. Next topic is mitochondria and chloroplast only. So in that also we can see so many ribosomes. Since it's a very important organelle, more amount of proteins are required. So these two organelles also have ribosomes. So that is about ribosomes, children. Next we'll go to mitochondria. Very simple organelle. Mitochondria. Mitochondria is a very important organelle because it is useful in ATP production and storage. ATP production and storage. You might be knowing what is an ATP molecule. ATP means adenosine triphosphate. It is the energy currency of our body. That means one adenosine molecule will be there. Phosphate, phosphate, phosphate. Okay. PO4 phosphate molecules are attached like this. Okay. So these phosphate molecules, when there is a breakage of this one bond, if this one bond breaks, it leads to 7,200 calories of energy will be released. So when ATP, 3 phosphates, is converted into ADP, 2 phosphates, it releases this much amount of energy. So our body stores energy and uses energy in this form. Whatever we are eating food, all that will go into our each and every cell. In each and every cell, it will be broken down, it converted into small molecules. Finally, this only will produce, ATP molecule is produced. So ATP molecule is nothing but energy. Where it is stored? In mitochondria. When our body needs energy, this bond will break. On this 7,200 calories of energy will be released. Depending upon our need, that energy will be released. If you are doing more work, more amount of ATP molecules. If you are doing less work, less amount of ATP molecules will convert into ADP. So this is the ATP production and storage occurs in the mitochondria. Next, let us see the structure of mitochondria. How many mitochondria do you think are present in one cell? From 100 to 150 mitochondria are present in each cell. So it is a numerous, one of the numerous organelle, one of the largest organelle that is present in plants, that is present in all living organisms, not plants, not animals. All the living organisms have this mitochondria. That means all eukaryotic cells, not prokaryotes. All eukaryotes have mitochondria. In prokaryotes, it's not present. We'll discuss about prokaryotes when we discuss about nucleus. Okay, what is the structure of this mitochondria? Mitochondria has two membranes like this. Two membranes are present. And this inner membrane has folds inside like this. It is folded. So, this outer membrane is outer mitochondrial membrane. This is inner mitochondrial membrane. In between there is intermembranous space. This inner membrane has so many folds. These folds are called as cristae. Over this cristae, we can see there are some particles that are connected to this like this. These particles are called as F1 particles. F1 particles. So they are very, very important particles for the synthesis of this ATP. They are very important for the production of this ATP molecules. So what is present in this matrix? Okay, this inside there is a jelly-like liquid that is present inside this. This part which is having this jelly-like liquid that is present inside the inner mitochondrial membrane is called as matrix. Inside this matrix, you can find a DNA molecule. DNA is present inside. Ribosomes are present. And you can find some starch molecules. Okay, carbohydrate molecules will write. So these are the components that are present inside this mitochondria. So mitochondria has outer membrane, inner membrane. Inner membrane have folds. Why do mitochondrial inner membrane have folds? If there is more fold, this Fn particles can be more. If more number of Fn particles are there, more ATP production. That is to increase the surface area. If surface area increases, more number of Fn particles. More number of Fn particles, more ATP production takes place. So that is why these folds are present. Okay. So this is cristae. On cristae, Fn particles are there in the spaces that is present in inner membrane. Matrix is present. Inside the matrix, you can find DNA, ribosomes, and some carbohydrate molecules. So that is it, children. Function, it makes the ATP molecules and stores them. Next one, organelles are plastids. Plastids or the color imparting substances that are present in the plants. Okay, the plastids are, there are three types of plastids: one, two, three. Leucoplast, second one is chloroplast, and third one is chromoplast. Leucoplasts are helpful in, they are white in color, as the name is saying, leuco, white. Plastids, their main function is storage of food. Storage of food. Wherever there is storage, you can find this leucoplast. Carrots have leucoplast in roots, and where the food is stored, wherever you can find some of the stem stores food, some of the root stored food, some of the fruits are storing food. These all storage tissues are, radish is also storing the food. Very good. Yogita, thank you. So in all those areas, you can find this leucoplast. Leucoplast store carbohydrates, lipids, and also proteins. Those carbohydrates which store the leucoplasts which store carbohydrates are called as amyloplasts. Those carbohydrates which store lipids are called as elaioplasts. Those carbohydrates which store proteins are called as eleuroplasts. Okay. Now, chloroplasts we'll discuss in detail. Chromoplasts, they give color to plants. Give color to plant parts. Like they are present in fruits and flowers. Fruits and flowers. What is their main function is to attract insects for pollination. They attract insects for pollination. That is their main function. Okay. Now we'll discuss about chloroplasts in detail. Chloroplasts have greens. They are green in color, as the name says. Why they are green in color? Because, can't hear me? They are green in color because they have a pigment called as chlorophyll. Okay, this chlorophyll imparts green color to this chloroplast. And coming to the structure of chloroplast, chloroplast has three membranes. Two of them cover the chloroplast like this. Two double membrane envelope. It is called as double membrane envelope. Third membrane is folded into structures like this. This is third membrane, which is folded into small coin-like structures called thylakoids. So the third membrane is folded into small coin-like structures, and this each one is called as thylakoids. The stacks of thylakoid is called as stroma. There are not only a single stroma, so many stromas are present depending upon the chloroplast. All chloroplasts do not have equal number of stroma. Depending upon the chloroplast, there are different numbers of stroma. These stroma are connected together by membranes. These are also membranes called as stroma lamellae. So stroma lamellae connects like this. Once one stroma is connected to another by the stacks, small membrane-like structures called stroma lamellae. Sorry, children. This stacks is called as grana. This is called as stroma. The liquid that is present inside this chloroplast is called as stroma. Sorry to confuse you. The liquid is called as stroma and the stack is called as grana. Okay? So, same like mitochondria, inside this chloroplast also, we can see the structures which are made up of some DNA molecule. This is DNA molecule, circular DNA, ribosomes are present, and you can see some starch molecules here and there. Chloroplast's main function is production of starch molecules. You can see some starch globules. So very simple structure. This outer two membranes is a double membrane envelope. Double membrane envelope. The third membrane is folded into thylakoids. The stacks of thylakoid are called as grana, and grana are connected together by stroma lamellae. This entire thing is present inside the liquid matrix called as stroma, and inside the stroma you can find DNA, ribosomes and some starch globules. Very simple structure of chloroplast. We'll see the function of chloroplast. The only function of chloroplast is it traps the sunlight, and converts the solar energy that is coming out of this into chemical energy. This chemical energy is nothing but starch molecule. See? So this is the function of chloroplast. It converts solar energy into chemical energy. In other words, simple words, it helps in photosynthesis. In plants, it plays the role of chloroplast is to produce food by the process of photosynthesis. Okay, children? So that is about chloroplasts. This centrioles is an organelle which is present in animals, absent in plants, absent in prokaryotes. But it is present in animals. Okay. In this you can see there is barrel-shaped structure, two barrel-shaped structures which are arranged like this at right angle to each other. Okay. This is one centriole. This is one centriole. Two centrioles. Okay. So that is why centrioles. We never say centriole. So these two are two centrioles. Centrioles. Okay. These centrioles are surrounded by a jelly-like structure. This jelly-like structure is called as centrosphere. Centrosphere and centrioles together is called as centrosome. Okay, this is an organelle which is not having membrane around it. Okay, ribosomes do not have membranes. This is the second organelle which is not having any membrane. Only this sticky jelly-like substance holds these two centrioles together and nothing else is there around this. If you see the TS of centrioles, you can find that there are 9 sets of 1, 2, 3, 4, 5, 6, 7, 8, 9. 9 sets of 3 fibers are present. This is one set, 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 this is one set. In the center, there is nothing. Gap. So this type of arrangement is called as 9+0 array. Every type of fibrous proteins, take it cilia, take it flagella, take it centrioles. Centrioles are also fibrous structures. Okay. So these structures usually have certain pattern of arrangements of fibers in them. Okay. Protein fibers are arranged in them and there is a pattern for the arrangement of fibers. It is different from flagella, it is different for cilia and it is different for centrioles. Okay. In centrioles, 9 sets of 3 are there. Okay. 1, 2, 3, 4, 5, 6, 7, 8, 9. So this is one set, this is one set, this is one set. 9 are there. In center there is nothing. If you take flagella, it is 9+2 arrangement. So it's different. It's 9+2 arrangement of fibers in it. That is about the structure of centrioles. What is the function of centrioles? What do they do? During cell division, this is a cell and every cell undergoes a cell division. Each cell will divide to form 2 cells. Again, each cell will divide to form 2 cells. This process is called as cell division and it is very important for our growth. Our body should grow, repair the tissues. Sometimes damage occurs. We are using the tissues continuously. Some of the tissues become old, they die. They should be replaced with new cells. Some cells like RBC, WBC, platelets, they die very frequently. They should be replaced. So that is the purpose why our body cells have the capacity to divide. Okay, it helps in growth and repair. So during this cell division process, what these centrioles do is, this is one cell. They move to the corners and they help in splitting of the nucleus into 2. Okay. So the centrioles during cell division, they move to poles and help in splitting nucleus into two. Nucleus into two. That is the main function of centrioles, right? After the cell division is completed, again they migrate to their position which is just beside the nucleus, like this they are. Whenever cell division should occur, they move to the corners and they help in dividing nucleus into two. Okay, so that is their main function. Normally, they are present just beside this nucleus like this in right angles. When cell division should occur, they move to corners and they help in dividing the nucleus into two. So that is about centrioles, children. So today we have discussed plastids, mitochondria, what else? Ribosomes and centrioles. In our coming class, we'll discuss about the remaining organelles. Right?